Yep, sounds good. Think there we go. Got my video started here. All right. So I'm going to start sharing my screen here. Let's see. All right, everyone see that okay? Very good. Well, thanks for having me to this crop school today. I'm sorry that I couldn't be there or that we all couldn't be there in person this week. I know it's a really great event that we've all come to love and enjoy. So it's disappointing that we have to do this remotely this year, but I appreciate seeing a large group of people here today, and hopefully we can still share and learn some things together. So I'm the extension specialist for soil fertility and nutrient management at Penn State, and I also dabble in a lot of issues related to soil health and carbon cycling and dynamics. So my presentation today is not so much about nutrient management. It's more about soil health and carbon management in the soils. So there's a lot of benefits and reasons that we should be interested in soil carbon, and soil carbon is very closely linked to a lot of soil health processes. So carbon is the major element in soil organic matter. It's about 50% of the weight by mass of soil organic matter is carbon. Carbon is the most abundant element in soil organic matter. And with that carbon and with that organic matter, we get increases in nutrient availability, increases in soil aggregation. That aggregation really helps to resist erosion and improve water infiltration into the soil, helps to create resilience against soil compaction, resilience to drought through increased water holding capacity, and back to the infiltration. When it does rain, more of that water gets into the soil as opposed to running off. And then resilience to excessive rain, and drought and excessive rain. Those are all things that we're sort of constantly oscillating between those two conditions in our changing climate now. So another reason to be interested in soil carbon is for carbon sequestration purposes. So there's an opportunity in agriculture to increase soil carbon levels through our management practices, and that's carbon that we are taking out of the atmosphere, carbon dioxide that we're taking out of the atmosphere and storing it in the soil. That can help reverse the trend in global warming. And there's new opportunities for farmers to participate in carbon markets where they actually get paid for the service of storing carbon in their soils. So what I want to do today is talk a little bit about the background of soil carbon management in agricultural systems, some of the processes that come into play, how we can manage them, and some tools that we have to predict what soil carbon levels might be under different management scenarios. So the first thing that we have to understand about soil carbon and soil organic matter is that in the last 10 to 15 years or so, our scientific understanding of what soil organic matter is and how it's processed in the soil has completely changed. So those of you that maybe learned about this in school more than 15 years ago, the universe has completely changed. And so I want to bring you up to speed on our current thinking about what soil organic matter actually is and how it's processed in the soil. So the old view of soil organic matter is that stable organic matter was formed from large molecules, things like humic acids or fulvic acids, that they accumulated in the soil because they were large molecules and they were resistant to decomposition by microbes because of their size and complexity. And so this is an example of a suggested structure for a humic acid. And one of the key features are these rings here. These are called aromatic rings. It's very difficult for microbes to break down these rings and just to have the enzymes available to break down these structures. But what we've learned in the last 10 years or so is that these humic acids and other types of large molecules in the soil, like fulvic acids in humans, actually do not exist under natural scenarios. These are actually products of the method used in the lab to measure organic matter. So they would use this kind of organic matter fractionation where they treat the soils with a very harsh alkali, and then they treat it with a very strong acid at pH 1.0 to sort of separate these different structures of organic matter in the soil. And what we found out is that actually these humic acids and these fulvic acids don't exist in the native soil organic matter. They are just a product of this treating with the alkali and the acids. These things sort of spontaneously are created in the laboratory through this fractionation process. They don't really exist in nature in the soils. So what actually does exist in nature in the soils are actually very simple biological molecules, sugars, proteins, amino acids, starches. These are sort of simple biomolecules that are the residues of microbial life, residues of crops. And what actually stabilizes and creates stable soil organic matter is not the size and complexity of the molecule, but actually an interaction with clay surfaces. So in this diagram here, the molecules are a little bit more simple in their shape and structure than what I showed you for a humic acid. And this is here in this diagram is the surface of a clay particle. And so it's through these interactions with the clay particles that organic matter becomes stabilized against decomposition by microbes. So there's a really important role that soil minerals and soil texture, particularly clay particles, play in this stabilization process. So I want to talk a little bit about, under this new view, processes that control the stabilization of carbon in the soil. And so I've created this kind of schematic diagram here that I'm going to step through and use this as a tool to help understand the processes that lead to carbon stabilization as well as carbon losses from the soil. So the first thing that we start with are inputs of carbon, and these inputs are crop residues, roots, manures. All of this material tends to be about 50% carbon by weight, sort of 40 to 50% carbon by weight. And these inputs, as they're added to the soil, they are consumed by microbes, and we call that process decomposition. And this circle here is the microbial biomass, and those microbes are made up of bacteria, fungi, actinomycetes, protozoas. There's a very rich microbial food web. I'm not going to get too much into the details of that food web, but the microbes do two things with the carbon that they're decomposing. One of the things they do is they grow their biomass, okay, and that's what's inside the circle here would just be the structures of their bodies, the cell walls, the enzymes, things like that they need to build their biomass. But they're also using that carbon as a food source to create energy to fuel their bodily functions. And the byproduct of that energy consumption is to respire carbon dioxide back into the atmosphere. So just like you and I, when we eat our breakfast, that gives us energy for the day. And throughout the day, we are breathing out carbon dioxide as we convert that food source into energy to fuel our activities. The exact same thing is happening with microbes. So this loss of CO2 through that respiration, that just goes back into the atmosphere, and it's not going to be assimilated into any type of soil organic matter that remains in the soil. So microbes, as they are doing their things, they are exuding glues and enzymes into the soil. They are also dying themselves. So, soil microbes don't live forever. They die and turn over. And so they have all of these microbial byproducts that can then interact with soil minerals and become stabilized. So in order for soil carbon or carbon to become stabilized in the soil, it has to go through this microbial processing, essentially. The raw inputs are not immediately stabilized. It's the microbial byproducts that become stabilized. Now, some of these byproducts, if they don't get stabilized, they can be utilized again by microbes. So microbes will eat dead microbes. Microbes will eat the glues that are produced by other microbes. So not everything that a microbe exudes or secretes or when it dies becomes stabilized, there is some further decomposition. And so we're going to talk about some processes that, maybe will prevent stabilization and promote more decomposition as we go through the presentation. So let's look at how much carbon is in each of these various pools. So in terms of the inputs, we call this particulate organic matter. It's sort of undecomposed residues and litter in the soil. It's about 15 to 20% of the total organic carbon pool in the soil is in these kind of undecomposed form. The microbial biomass is about 1 to 3% of the total carbon, and then what's stabilized on the surfaces of minerals is about 80% of the total carbon. So the vast majority of the carbon in the soil is what's stabilized on these mineral surfaces here. Now, if we were to convert these percentages into actual quantities or masses of carbon, if a soil had 1% carbon or about 2% organic matter, so organic matter is about 50% carbon, there would be about 20,000 pounds of carbon in an acre furrow slice of soil. Remember, an acre furrow slice is about 2 million pounds of soil. So 1% of that being carbon would be about 20,000 pounds of carbon. And then if we, say 15 to 20% is in the particulate form, that's about 3,900 pounds of carbon per acre. This is about a year or year and a half's worth of crop residues from an agricultural system. So imagine you're adding all of that residue to this pool. Over time, you kind of build up an average level of about 1 year's worth of crop residue in the soil in this particulate organic matter fraction. Microbial biomass is about 200 pounds carbon per acre. And this is one that always surprises me how little this is, because you hear people talk about their underground herds of microbes. You've got a herd of livestock in your soil that's doing all these great things. Well, you don't have a herd of cattle by any means. You have maybe a couple dozen chickens or so there in the soil in terms of their weight of microbial biomass. And then what we have in terms of mineral associated organic matter, vast majority, about 1,600 pounds carbon per acre. So let's talk about some of the factors that can affect the stabilization of carbon as we move through these pathways to stabilization, or potentially not stabilization if we're respiring it. So the first of all would be the quantity of carbon inputs, and generally, assuming other limiting factors don't arise to stabilization, the more carbon inputs you have, the more you're going to grow this microbial biomass, the more byproducts are going to be created, the more byproducts will become stabilized on the surfaces. So we can increase stabilization in soil carbon by adding more crop residues, growing cover crops, adding manures to the soil, retaining crop residues. Things like that should increase soil carbon levels. The other thing we can think about that changes stabilization is the microbial biomass. How much of the carbon did they utilize to grow their biomass versus how much did they respire? And we call that the microbial carbon use efficiency. It's the proportion of what they decompose that grows their biomass, which will then create byproducts that can be stabilized, versus how much they respire. And the more they respire, the less that will remain as byproducts to be stabilized. And one of the interesting things about the microbial communities is that fungi have a greater carbon use efficiency. So fungi will create more biomass and respire less carbon dioxide than bacteria will. So one of the things you can do from a management perspective is to try and promote more fungal biomass as opposed to bacterial biomass. And one of the things that does that is no-till. So fungi are very sensitive to tillage and disturbance of the soil that mixes up their mycelium and hyphae and whatnot. So you tend to get more bacteria. So one of the ways that no-till could potentially help increase carbon sequestration would be through promoting the fungal biomass in the soil and having less of the carbon that's decomposed respired as CO2. The other thing that can affect carbon stabilization is the quality of the residue inputs. And so high quality residues are usually more efficiently processed into microbial biomass, and less is respired as CO2. So the quality of a residue, how easy it is to for the microbes to decompose the easier it is for them to create biomass out of it, the less work they have to do, the less carbon dioxide will be respired. So here are some examples of high quality residues, things like carbohydrates, proteins. These types of molecules are very quickly assimilated into the microbial biomass, and the microbes have to spend relatively little energy to decompose those, so there's less CO2 respiration. So things like legumes, fresh manures, root exudates are all things that are considered high quality residues. Lower quality residues would be things like lignins, polyphenols. So these types of molecules can be broken down by microbes, but it takes them more energy. So they're producing more CO2, consuming energy to build their biomass from these types of molecules. So you get less byproducts and less stabilization. So things like wheat straw, corn fodder, cover crops that you let to grow to be too mature, composted manures versus fresh manures, these are all materials that microbes need to spend more energy breaking down, and so there's less that remains in their biomass and as byproducts for stabilization. And that's kind of counterintuitive because there's a lot of people that want to grow bigger cover crops, more mature cover crops to create more biomass. There's a little bit of a cost to that, in that those residues will be less efficiently processed by microbes into stable organic matter. So another factor that can affect carbon stabilization is the temperature. And as soil temperatures increase, microbes respire more. So just to stay alive, or just their metabolic activity increases with soil temperature. And so they will end up having more CO2 respiration, less microbial biomass and byproducts. So in warmer temperatures, we tend to see less soil carbon accumulating than we do in colder temperatures. So the climate zone that you're in will affect your soil organic matter. Also management. So no-till, keeping a residue cover on the soil surface, that will keep the soils cooler, and theoretically that should allow for less respiration and more stabilization. And then we've got soil texture as a property that regulates carbon stabilization. So the amount of mineral surface area that you have, the more clay minerals you have, the more surface area you'll have of mineral surfaces, the more stabilization that can take place. So, typically as the clay content of the soil increases, if you hold everything else constant, the organic matter level or the soil carbon level tends to increase because of that increased stabilization. So this is just a figure of soil samples that I've collected in the last few years from the Rock Springs Research Farm. So it's all the same climate. They've all been grain rotations with no manure additions to them in relatively recent history. And plotting the clay content of the soil against the percent organic matter, we see this increasing trend where going from 20% clay to 50% clay, we have gained about 1.5 points of organic matter simply because of that change in soil texture. And soil texture here, the clay content, explains about 30% of the variation in organic matter levels across this data set. So we've talked about a couple different things that no-till systems have in their advantage for carbon stabilization, so increasing the fungal biomass, reducing soil temperatures through the mulching effect. But one of the challenges that no-till systems are running up against, in my opinion, and analysis of the data, is that we've got something going on called carbon saturation. And essentially what is happening is we are not mixing the carbon throughout the whole plow layer. It's remaining at the soil surface, and it's interacting with a very narrow zone of soil, and that soil, the minerals in that surface soil are becoming saturated. So their surfaces are saturated with organic matter, and there's no more room for additional stabilization on the clay surfaces. And what happens in that case is the microbial byproducts do not get stabilized, and they just undergo further decomposition and carbon dioxide release into the atmosphere. And so you can increase your inputs all you want, but the pathway from the byproducts to stabilization is basically closed because of saturation, and those increased inputs just recycle themselves and ultimately get respired back into the atmosphere. So this is kind of a compound problem of carbon stratification in a narrow zone of soil at the surface, as well as saturation, these two things interacting with each other. And this is data from an on-farm soil sample collection that we did in central Pennsylvania. It's a dairy farm. They've got a corn alfalfa rotation. They've been no-till since the year 2000. They apply 6,000 gallons per acre of manure in the fall and the spring, and they grow a rye cover crop after their corn silage, and sometimes harvest that rye cover crop for forage, depending on the year. But when we soil sampled by depth increments, so we had a 0 to 5 centimeter soil sample, a 5 to 10 centimeter soil sample, 10 to 20 centimeter soil sample, and we look at the soil carbon level in each layer of soil, and across 4 different fields, this is very consistent. We see the soil carbon levels dropping off quite a bit with depth because that carbon is not getting mixed throughout the whole soil profile like it would be in a tillage-based system. And so we see that 10 to 20 centimeter zone of soil has about half as much carbon in it as the 0 to 5 centimeter soil does. So this is that effect of carbon stratification caused by the no-till system. So one of the things we wanted to explore, kind of an idea, is could there be some maybe benefit to occasionally tilling a no-till soil, maybe once every 6 years or so? And there's a lot of farmers and a lot of consultants where this idea is just dead in the water because understandably they're a never tiller, and because they want to preserve all those benefits of no-till. And to some extent, I agree with trying to limit tillage as much as possible because of all the benefits that system brings. But there is a challenge in terms of maximizing carbon sequestration that we're running up against when we don't ever till the soil. So what we wanted to do is actually use a computer model of carbon dynamics to test the idea that maybe tilling the soil once every 6 years or so, you could sort of reset that stratification and alleviate the carbon saturation effect in the topsoil by occasionally mixing the soil, bringing that really high carbon surface soil down a little bit, bringing the lower carbon soil up a little bit, just mix things back up and take care of that stratification and alleviate some of the carbon saturation. So we did a simulation using a model called the cycles model, and in a second I'm going to demonstrate an online website that we have that can run this model for anyone. It's publicly available, and I'm going to do a quick demo of it. We ran this model and we compared the farmer's management practice. These are the 4 fields I just showed you before of no-till since 2000. Then we ran from 1980 onward a continuous tillage, a continuous no-till, or tillage once every 6 years. And I'm just going to focus on the first field here. But what we saw is that basically there was an increase in overall percent carbon in the soil, and this is just for the topsoil here, by doing tillage once every 6 years. Basically, we ended up having maybe about 0.2% points more carbon compared to the farmer's management of doing no-till since 2000. And actually, if they had been no-till since 1980, it looks like their soil carbon would have been even lower. So that was actually kind of a peak, and then it starts to deplete in terms of soil carbon. So what I want to do here is shift to our website, our Cycles website, and show you how it can be used to run simulations. So Let's see here. So the cycles model it's at this website here, and I put that in the slides. You can kind of jot this down if you want. When you log in, so I'm already logged in, but you can get a login. You create a user account and password. There's no fee for this or anything. It's freely available through grant funds that have made this happen. When you log in, you see a map of the United States, and you actually can do a simulation anywhere in the continental United States. We have all of the data in terms of soils and weathers and whatnot available for the whole United States. I'm going to zoom in to Pennsylvania here, and as you zoom in on the map, so the information that shows sort of starts to change. You get more details on the roads. And eventually you start to see farm parcels show up, and eventually fields, and then soil types eventually will show up. So here are the dotted lines that indicate different soil types. And so to start a simulation, you click on this little button up here that says select field by point, and you just drop a point, and that will start the simulation. So I'm going to just start a simulation here. You can set the time frame that you want to run the simulation. I'm just going to run a 2-year crop rotation here, so I'm just going to enter 2-year simulation. But what we do is we actually run this simulation to sort of infinity. So at this point, we're just setting how many years do we want in our rotation. I'm just going to set a 2-year simulation here. You click Add Simulation, and then over here, there are some little menus that we can go down. One of the things I'm going to do is I'm going to create automatic nitrogen so that my crops are fertilized automatically. They don't experience any nitrogen limitation. But we've got soil information about the soil type where I dropped this point. So this is just from the NRCS database. We have layers of the soil profile with their sand and clay content and organic matter content. There's a weather file that looks up weather back to 1980. And then the next step is we create basically a management scenario. And so that we do that on the timeline. And so I'm going to create what's called a rotation group. I'm just going to call it corn soy rotation. Started in 2018, ended in 2019. We'll create that on the timeline. You click fit. And then what you do is you kind of drop in the crops that you're going to be growing. You can move the little bar here. You can plant a crop. There's a number of different crops that are preloaded in here to choose from. I'm just going to plant a corn grain crop here. And so that's going to plant corn. I'm going to move to May of the next year and plant full season soybean crop. And now I have a crop rotation in here. And the next step would be to then submit this crop rotation to the model. So we go to the cycles button here. We click submit task, and it reads in the weather, the soil type, the management information. And we can then click on the results tab to look at changes in soil carbon. So I'm going to look at the soil carbon results here. And this shows me a soil profile for that corn, soy rotation, different layers of the soil, and 2 different years of the rotation, the carbon levels that would accumulate throughout the whole profile here. So if you wanted to go back and then add, say, a poultry litter application to the corn, you can do that with a fertilization option here. We've got manure as one of the options. I'll just put 3 tons to the acre, 70% dry matter, about 15 pounds ammonium, 40 of organic N. Fill that in and save that. I can then come back and submit that new management scenario and evaluate the results. We will look at the carbon results here, and you can evaluate what the new outcome is of that. So, this is a tool that is available now, and we actually have funds through Northeast SARE to run some trainings on how to use this tool. So if this looks like it might be something interesting for you to learn about for the work that you do with farmers, then when you take the final or when you take the survey, evaluation survey about this presentation, there's a question that says, are you interested in attending a training about the cycles model? If this interests you, just click yes to that, and we'll gather your information and let you know when we host that training. So I'm going to shift back to the my slides here and talk a little bit about some results from using the cycles tool to test different management scenarios and their carbon sequestration potential. So I ran these scenarios on Hagerstown soil in central Pennsylvania, a corn, just a continuous corn rotation with residue returned to the soil. We accumulated 59.6 megagrams per hectare of carbon. I tried out this occasional tillage once every 6 years, and it only boosted carbon levels by about 1 megagram per hectare across the whole soil profile. So we're talking about a mass of carbon here, not a percent of carbon. So there was just a very modest gain when we did the corn with continuous tillage every 6 years. When we added a cover crop to it, very, very modest gain in carbon additions from the cover crop there. Again, with the cover crop and doing tillage once every 6 years, we only had a slightly modest gain in the carbon that was stored. Now, when we added poultry litter at 3 tons per acre for every year of the corn, so basically every year we're adding 3 tons per acre of poultry litter, this gave us a really big increase in our soil carbon because of that addition of the poultry litter. We went from 60 to 78.2. And then when we incorporated poultry litter with tillage once every 6 years, this is when we saw our biggest gain in total carbon storage. So this tillage every 6 years, it's something that probably you don't need to do unless you're reaching some carbon saturation level through really high inputs of organic amendments through the poultry litter. So this big gain from the tillage once every 6 years, we didn't see anywhere near that kind of gain when we were just cover cropping or just corn itself. It was only when we had the manure mixed in. So there's some interaction there. This is what the soil profile outputs looked like. So without doing any tillage, we see that our poultry litter accumulates to our top layer of soil, the 0 to 5 centimeter, accumulates to a very high percent carbon level, and then it drops off very quickly because of lack of mixing. Once we start doing that tillage once every 6 years, we see that carbon's getting mixed into the soil profile where it can be more effectively stabilized. So here we've basically reached the carbon saturation potential of the mineral surfaces in that 0 to 5 centimeter. But doing an occasional mixing really helped increase the soil carbon storage. We can look at a corn-soy rotation, and the interesting thing about this is that soy is going to be a higher quality residue to be incorporated than the corn fodder is. So shifting to the corn-soy rotation under no-till management, saw a very slight increase, maybe 1 megagram per hectare. But when we mix that corn-soy rotation with doing tillage once every 6 years, we saw a big boost. So this is another aspect where there's an interaction between that occasional tillage and the rotation. Adding a cover crop, modest gains in soil carbon, but corn-soy rotation plus cover crop plus tillage every 6 years, big boost. And that's from the more effective stabilization of that high quality residue from the soybeans. And so, adding poultry litter, we saw a boost there, an occasional tillage with poultry litter every 6 years, big boost in soil carbon. So again, these are model simulation results. We do have active research to try and verify, is this behavior true? Because if it is, it has really important implications for the potential for carbon storage. So at this point, this is more of a thought experiment, but it's something that I think There's a lot of sort of fundamental theory to suggest that this might be true, and I think we can do more research to verify it. So I want to shift with the remainder of my presentation here into talking about different ways that soil carbon can be measured in the soil. And there's lots of different ways to measure different pools of soil carbon. And a lot of these different measurements are integrated into soil health testing services. So the Cornell Soil Health Test is probably one of the most well-known ones. Woods End Lab also has what they call the Solvita suite of analyses. But there's a lot of standard soil test labs that also will measure either total carbon or total organic matter as an indicator of soil carbon in the soil. So, these different soil test services, soil organic matter, total carbon, there's something called permanganate oxidizable carbon and microbial respiration are part of the different soil health testing packages depending on what level you're spending. Now, these can be pretty pricey, $70, $60, $110, $80 for these suite of assays. Sometimes you can get individual things done at standard test labs for much cheaper than running a full soil health analysis. So soil organic matter is probably the one that most of you are the most familiar with. This is typically done by loss on ignition, by heating a soil sample up in a muffle furnace. Temperatures vary between labs, but they might be from anywhere from 350 to 500 degrees centigrade. The amount of time that is spent in these ovens also varies between labs. But basically what's happening is the organic matter is burning off. This is a sample before it was put in the furnace, and after you can see it's lost a little bit of its dark color from the loss of that organic matter through the ignition. And they simply weigh the soil sample before and after ignition and see what the mass loss is. Now, some labs report the results on a raw percent loss on ignition basis. Other labs actually have a conversion where they convert that loss on ignition back to a historical method of measuring organic matter called the Walkley-Black method. And when you make that back conversion, you lose about a percentage point of organic matter value when you make that conversion. So you need to be really careful. Is the lab measuring it and reporting it as percent organic matter? Or are they measuring it, reporting it as percent loss on ignition? And when we compared Ag Analytical Lab at Penn State percent organic matter versus the Woods End percent loss on ignition, we saw the loss on ignition was about 0.8 percentage points higher. So this could be very confusing if you're sending soil samples to different labs and getting results back, and all of a sudden it looks like you've increased your organic matter by 0.8 percentage points. It might just be differences between labs. We also saw differences even in two labs that report on a percent organic matter converted back to the Walkley-Black method between Penn State Ag Analytical Lab and the Cornell Soil Health Lab. Very tight relationship, but there's this intercept, and this difference in slope and intercept at about 4% organic matter at Penn State. We're getting about 4.5% organic matter at Cornell. And this might be due to differences in their oven temperature. They run at a higher oven temperature than Penn State does. So I think this is something where consistency of labs and tracking things over time is really important to avoid sort of biases from different labs. Now, another way to measure carbon is, instead of measuring the percent organic matter, to just measure the elemental carbon. And we do this with a combustion analyzer. We heat the soil sample up to a very high temperature. But then instead of just measuring the mass lost, what we actually measure is there's a stream of carrier gas that measures the combustion gases through a detector, and we detect how much CO2 is emitted from burning that soil sample. And that CO2 is derived from the carbon in the soil. So this is a more expensive method to measure soil carbon, but it's highly accurate, it's highly definable what it is, and it's a very precise method. Now, the disadvantage is that total soil carbon changes slowly with time, so it's not that sensitive to management changes. Now, another type of measurement that can be done in the lab, and this measurement is called permanganate oxidizable carbon or active carbon. This is actually one of the answers to one of your quiz questions after the presentation to get your credits. It's basically done by shaking your soil sample with a potassium permanganate solution. So this purple solution is potassium permanganate, and that permanganate oxidizes a more active fraction of the organic matter in the soil. So it's an intermediately labile fraction of soil carbon. It's sort of a mix of a little bit of particulate organic matter, a little bit of microbial biomass, a little bit of microbial byproducts, but it tends to be correlated to microbial biomass. And the key here is it's sensitive to short-term changes in soil management. So within a year or two of implementing some management change, you'll start to see the direction of that change come out in these active carbon results. Now, this test is only available through the Cornell Soil Health Test. It's not available through any commercial labs that I'm aware of, or like the Woods End Soil Health Test is not running this potassium permanganate, only available through Cornell lab. And again, this is an answer to one of your quiz questions. Now, there is a good relationship between permanganate oxidizable carbon and percent organic matter. There's a positive increasing trend here, but also a lot of scatter around the line. And some of that might be due to the differences between the ability for this tool to detect a short-term change in management versus total organic matter takes a longer period of time to detect a change in management. Now, another test that's become quite popular is the microbial respiration test. Typically what they do is they take a dried soil sample, they re-wet it, put it in a jar, and measure how much carbon dioxide is released in a period of time. This is related to the size of the microbial population. It's related to the amount of organic matter that's available to be decomposed. It's a potential indicator of nitrogen mineralization. But one of the real challenges with this test is that it's really difficult to tell whether this test is measuring soil building processes versus soil depleting processes, because what we're actually measuring is the rate of carbon loss from the soil when you re-wet it due to microbial activity. That might be an indication of actually your soil is losing carbon if you're having more losses from microbial activity versus more stabilization. So this CO2 burst, or the CO2 respiration, the microbial respiration, is related to the amount of organic matter that is in the soil as well. So increasing levels of organic matter tend to have higher biological activity and more microbial respiration. Now, the last type of measurement that I'm going to talk about is called particulate organic matter. And this is actually not offered through any soil health tests or soil labs, but it's something that you could very easily do yourself. With particulate organic matter, what we're sort of measuring are the fragments of non to semi-decomposed organic matter inputs. We do this by sieving out the sand size fraction of the soil, just with a sieve like this. And this is what that fraction looks like. You can see little under a dissecting microscope. So this is magnified about 5 times. You can see the fragments of plant debris. You can see the grains of sand here. There's some weed seeds in here as well. You can also do this just by floating the organic matter. So the organic matter floats and soil tends to settle when you shake it up. And we've added a little bit of calcium chloride to this solution to just help the soil flocculate and settle. But these are 3 different soils that have increasing levels to decreasing levels of POM in them based on their management. So to conclude, there's a lot of factors that regulate soil carbon dynamics. Sometimes these factors work against each other, and might result in counterintuitive outcomes for management, such as maybe this occasional tillage strategy. And maybe having legumes in rotation. Even though legumes produce less residue overall, that residue is higher quality and is more likely to be stabilized than a low quality corn fodder. So there's lots of different ways to measure soil carbon too. Some of the challenges are the cost of these methods as well as interpretation, but I think just measuring total organic matter, maybe total carbon, is a great way to sort of get started at monitoring this. And I think in general, we have lots of opportunities to improve on our soil carbon management as we have new understandings of the dynamics and how management interacts with those dynamics. So I just want to recognize the Northeast SARE project that has funded some of this work, and we hope to have future trainings to look at the cycles model and delve into more aspects of that. So looks like there's maybe about 6 minutes left for questions, so I will turn it back to the moderator to moderate those questions.
Thank you, Dr. White. So yes, you have, looks like 9 questions. And just for your curiosity, you have 190 participants so far.
Excellent.
So questions, you have several questions related to tillage, and I'll try and summarize those all into maybe 1 or 2 questions. One is why the 6-year frequency related to tillage, and the other is related to the type of tillage. Can you explain a little bit on both of those issues?
Sure, sure. So 6 years was kind of an arbitrary frequency that I chose just to run these simulations. The dairy farm that we were looking at has a 6-year rotation and of alternating corn silage and alfalfa, so that you could sort of figure at some point in that rotation, either for maybe seeding the alfalfa or for coming out of alfalfa into corn, there could be some strategic opportunity for tillage at that point in the 6-year rotation. But I think there's opportunity to sort of figure out what is the optimum frequency. Is it once every 6 years? Is it once every 10 years? Because, certainly that tillage event might do some damage to soil structure, might create some erosion, potentially, it'll disrupt the fungal biomass, things like that. So we don't want to do it more often than we need to. So I think there's some opportunity. 6 years is not a magic number by any means. It was just what we used. Now, type of tillage, these simulations, what I simulated was a mold board plowing event, because that kind of maximizes the mixing of soil throughout the profile. And that would be the goal of these occasional tillage events, is to do as much mixing and redistribution of that carbon as possible to offset the stratification and saturation. So it's not the kind of thing where you'd want to go in with a light vertical tillage tool or even a conservation tillage tool, because the point is actually to mix the soil as much as possible. So it's a different strategy. So yeah, next question if we have time.
All right, so I think that answers the tillage questions. We'll start back up at the top. To what extent does the addition of commercial folic and humic acid increase CEC and provide some effect of organic matter?
Yeah, so there's a lot of products that are sort of humates or humic acids, and generally my feeling is that if you think back to that slide of how much carbon is there in a soil, and stabilized carbon, there's about 15,000 pounds per acre of stabilized carbon already in the soil. So you've got to ask yourselves, is the amendments I'm purchasing and applying, is it coming anywhere near to supplying 16,000 pounds per acre of carbon? And usually it's not. And so I think those products are a total drop in the bucket of what's already there, and probably highly ineffective at changing anything about the carbon dynamics and the cation exchange capacity that carbon provides.
Very good. I think we'll make this the last question that I'll pass on to you. There's other questions in the query here. I would suggest those folks reach directly out to you by email to ask those questions. But the last question I'll ask today is the driver of soil manure quality, the CN ratio. A fresh manure would be higher quality residue if it did not include bedding versus most applied manure that has some sort of bedding.
Yeah, so that quality of the residues is about the C to N ratio, and the lower the C to N ratio, basically, the more available that is, and the more quickly that will be utilized by microbes. When there's a lot of bedding mixed in, the microbes actually, in order to utilize the nitrogen, they actually have to respire that carbon off into the atmosphere. So there is a relationship with the C to N ratio of the manure. And so having a lower C to N ratio, more nitrogen, less bedding mixed in, should create a more effective stabilization pathway. Yep.
All right, so I think we'll stop there with the questions. And just to remind folks about if you're needing the CEU credits, I think there you go. There's a QR code.